All right, so I'd love to introduce Dr. Leo Wang from the University of Washington in Seattle. He came a long way to be with us today. He's going to talk about current clinical research and clinical trials for FSHD. Dr. Wang is a professor in neurology at the University of Washington. He's led the successful recruitment and clinical evaluation efforts as the co-investigator in the Seattle Wellstone Center. So please welcome Dr. Leo Wang.
Well, thanks for having me. I am from University of Washington, just south of the border, but I am Canadian. And so it's great to be back and thanks for having me. I was going to talk about some of the research that we've done at the University of Washington and some of the research that we're planning on doing and also talk about how this, I feel, plays the groundwork for some of the clinical trials that are going on right now and just giving you guys a little bit more about that information. I just have some quick financial disclosures, but none of it is really affecting how I would interpret this talk. A lot of the work that we're talking about is from the Senator Seattle Wellstone Muscular Dystrophy Specialized Research Center. So this is part of the U.S. Congressional Act, the ND CARE Act of 2001, that was reestablished and subsequent times to have 6 NIH-sponsored centers that is funded by the NIH, and we at the University of Washington have been really lucky to be part of this since 2014 until now. And we've had partners with Kansas University as well as University of Rochester. There's mainly 2 projects, and one of the projects that I was going to talk about is some of the stuff that we've learned for the translational research part. And that because it's focused on FSHD. These are our friends and collaborators and leaders who have helped us through these studies. And so the first part I wanted to talk about is really some of the stuff in terms of measuring progression by muscle and mind. Since part of the work that we did is the first Wellstone that started in 2015. So for that Seattle Wellstone project, we had 32 patients that we followed up at baseline and also a month later. We did the MRI of patients' legs and we measured those muscles that I showed there. And we measured how much muscle and how much disease muscle was there. And we looked at 512 muscles total over the 32 patients following them a year at a time. And so this is what we're trying to look for when we're looking for a muscle MRI. This is the muscle MRI for the leg there, and we're trying to see what parts of the muscle is still muscle and what's part of the muscle that's been having problems and has been converted into fat. And that is just a sign that the muscle is not healthy and is having trouble. And this is interesting in that it allows us to follow this over time and it allows us to quantitate, it allows us to see how the muscle is changing. And hopefully in the clinical trial, we can see that it can stop or it can even be reversed. And so the next slide is just an example of a patient that we follow along for over 4 years. And you can see that in the top left part, it's in 2012, and you can look at it clockwise and see how it changes over time. You can see the amount of muscle and the amount of muscle that has been converted into fat, and you can see that over time. And when we looked at our patients, we basically looked at 512 muscles and looking at them on the x-axis is all the muscles rank from the least amount of being affected to the most affected on your right-hand side. And so those patients are about, you can see that muscle has 90% of it been replaced by fat, while on the left-hand side, these are muscles that are less than 10% affected by fat. And I think the things that you learn is that, a lot of muscles, more than half the muscles are not affected or less than 10% replaced by fat. There are some muscles that are more affected. And when we look at how this changed over time, and the next slide, the yellow bars, or it's not really clear with yellow here, but each muscle, we're tracking how they changed over time. And I think what we're trying to show is that it seems as though the changes were really in the muscles that were about 10% to 50% faster flip. These are the muscles that are kind of in the transition zone. Those are the muscles that change every year, and that can be helpful tracking this in a clinical trial. And that's the next slide. It shows that on the x-axis is the number of, these are the muscles that were less than 10% affected or greater than 80 to 90% affected. You can see that change over time, about 63% muscles and the 40 to 50% replaced areas were progressing by greater than 10% over a year. And so this is kind of the work that laid the foundation for people to use MRI to measure changes over time in a clinical trial. The next part that I wanted to talk about is measuring was this reachable workspace that I think some of you guys have heard about, and it's been employed in multiple clinical trials thus far. And this was part of the work and part of the larger international consortium led by Kansas University as well as University of Rochester, and we were just fortunate to be part of it. And it's a NIH funded study that looks at clinical trial readiness to solve barriers to drug development in FSHD, and it's a 2-year study that looked at patients multiple times over those 2 years and seeing how they were changing. And so I was really going to talk a little bit more about what we've learned in the baseline visit to visit number 1 in terms of the functional outcome measures and reachable workspace that was done. This is part of a large network that's international, and we're part of that. We're very glad to be part of it. This is part of the FSHD Clinical Trial Readiness Network that is sponsored by multiple organizations, including the NIH and FSHD Society, and we're grateful for that. You can see on the screen that there are multiple centers here in Canada as well as the United States, and I believe that there's 3 in Canada and they're coming online at different times. You can also see that there's a few centers in Europe as well, and also in Brazil and Australia, which is helping the pediatric FSHD part of these studies. So most of these centers, a lot of these centers gathered information in the Resolve clinical trial, that first trial that I was talking about. And one of the things that I'm trying to understand is the reachable workspace, and is if that was something that can measure how much the patient is able to reach in front of them, and whether that could be tracked over time. And so what this reachable workspace is doing is in the next slide. So you're sitting in front of the large screen in that camera. Then our patient is on the top right. They're supposed to follow the screen. This is what you're showing in front of the screen. Tells you specifically what kind of maneuvers that you're supposed to do. And then on the bottom right is basically what the computer program is trying to measure. Is how much are you able to gain in that area in front of you. They're very specific movements that we're looking at. And get across your bottom towards your arms. You bend your elbow. You then go back and bend the motion as far as it goes. Next, you arch straight, so bring your hands to your shoulder level and move it across the bottom towards your opposite shoulder. You bend your elbow. Then go back and move it as far as it goes. Then lift your palm above your shoulder level and move it across your body. Now, with your palm facing your left, down facing forward, stretch your arm and hand you as far as it goes. So what this measurement is able to do is basically like treat what you can reach as a sphere for one of your arms and see how much of that, what percentage of that can you reach. So this is what you can reach for that patient, and this is what a control patient will look like. It's not perfect, but you can see how much it is it's reached. And so what you're trying to measure is you're trying to measure on the right side and trying to measure on the left side, and you have these different quadrants in front of you that you divide to, and you try to measure how much total that can be measured. And what we did as part of this international consortium was comparing it to the quantitative muscle testing. And so having this this gizmo that basically puts on straps at different parts and are able to measure the strength of your deltoid, the strength of your biceps, the strength of your triceps, and your strength rotating out your arm. We were trying to see, does this reachable workspace correlate with how much strength you have? And so what we found is on the what we found was that it was a decent test of if you could reach quite a bit, and that's if you go towards the right on both of these plots, you can see that there's more strength that you can get. That's on the y-axis. So the more you can reach is definitely correlated with the more strength you have in your arms, and this would be helpful in saying that what you're measuring is probably something that is important, is probably related to the muscle. And so then the next question is, in the last few minutes, I was going to talk about can we use these muscle MRI measurements? Can we use reachable workspace to measure change in the clinical trial? And I don't know if you guys have heard about this, but it was just a clinical trial that finished. It was unfortunately unsuccessful, and I feel really proud of it. We know that there are patients here today who are part of it, and I think we learned a lot from it. And so I wanted to talk a little bit more about it, and also about how that study really put the foundation of how to use muscle MRI and how to use reachable workspace to measure change in future clinical trials. So this was the losmapimod phase 3 trial. It was 48 weeks. It was about 260 patients, and there was a placebo arm and there was a treatment arm for losmapimod that you would take twice a day. And there was data for the MRI and the reachable workspace at the baseline, and there was MRI and reachable workspace data after 48 weeks. So this was the results. So the losmapimod phase 3 trial, 48 weeks, reachable workspace results. What you're seeing on the x-axis is time. So we have baseline 4, 12, 24, 36, 48. And then on the y-axis, how much was changed. And we're looking at the reachable workspace. So how much you can reach in front of you in the dominant arm. And you're looking at it over the time. And I think the things that they found was there was no difference between the placebo arm, which is blue, and the treatment arm, which is orange, at 48 weeks. You could see that patients got better. And so it seemed as though being part of the clinical trial gave a lot of patients also some confidence to start doing exercises, or they may have learned a little bit more how to do the tests. But it seems as though one is that patients who were getting better, it's a small amount, but it was there. It showed the error bars are actually pretty good, they're pretty tight, and it actually was able to I think quickly tell the company what the results were. And this was comparing that 48 weeks. There was really no difference between the average total RSA. So the average of both arms, how much space you can get, the percentage of space you can get, average per patient. There was no difference between the orange treatment arm and the blue placebo arm. And so what about the MRI results? So this is the results from the phase 3 trial at 48 weeks. So on the x-axis, you can see the data from the baseline, the week 4, and then basically a year later at week 48. And you can see that on the y-axis is how much change in the muscle was there over time. And so you can see that over time, there was a little bit of change. It was really like 0.2, 0.4. So small changes over about a year. We can see that the placebo arm got a little bit worse, and then the losmapimod arm was not as bad it was not statistically significant, so when we ran the statistics at 48 weeks, it wasn't, you couldn't show that there was a difference, but you can see that there is some separation in that, and so this might suggest that, you know, the MRI is telling something, and further analysis of the MRI results may give us may give us other clues about what's going on. I think that the thing that one would take away from this is that maybe the trial was too short. Maybe 48 weeks was too short. Maybe you need to look at a year and a half. Maybe you need to look at 2 years, and maybe you can see the difference. But the difference probably wasn't going to be spectacular. It wasn't going to be a huge change, but it could be a significant change. So if you extract it out, maybe over 2 years, maybe the difference was going to be 0.5 versus 0.8. Maybe that's what the difference is. And so that's something to think about. So this was the data over 48 weeks. You know, over 48 weeks, multiple small command, the muscles gained about 4%, and specific muscles that we're looking at, and the amount of conversion from muscle to unhealthy muscle, it was 0.6 in the placebo arm. So it wasn't a statistically significant difference, but it was a slight difference, and maybe time is the issue. And so these are things I think builds the foundation upon multiple natural history studies, and also clinical trials to understand how you measure these things, in a larger population that changes over time. We're doing further studies of this as part of the clinical trial readiness network. And this is the MOVE FSHD study. It's a prospective observational study, and it's looking at 450 participants. It's looking at it throughout the United States, Europe, and also Canada. There's 12 sites in the US, and there's 3 planned sites or recruiting sites here. And we're trying to follow missions over 3 years, and the visits are hoping to occur annually. We're also trying to do more in terms of understanding the MRI. So there's a sub-study called MOVE Plus, and it is looking at 200 patients who are part of the movement. So it's a sub-study, and it's looking at patients who can walk 30 feet without support, and there's no contraindications for MRI, and that's what we're trying to do. There are newer techniques to look at the MRI that is able to look at the whole body and to see how those change over time, and so you can get a lot more data, because the FSHD affects different muscles differently, and so this is able to follow all the muscles over time for a patient. The additional biomarkers, the additional things that we're trying to collect are the reachable workspace, blood, and muscle tissue. Right across the border, we're also trying to do the clenbuterol study. Clenbuterol has been shown to decrease sport, and that's the transcription factor that gets turned on in muscles with FSHD, and it gets turned off in cultured muscle cells from patients. This drug has been approved in Europe for the treatment of asthma, and it's been used in bodybuilders, muscle growth. We're trying to do an open label 6-month study to look at clenbuterol on muscle, and so on a muscle biopsy, and also on a muscle MRI. And hopefully the study will continue if the United States government allows the funding. So we'll see how that goes. Is that for me? Oh, okay.
That was me.
So better understanding of the molecular basis of FSHD, there's been a lot of work done, a lot of scientists and a lot of thinking about this. Treatments have been designed to target at the molecular level, and I think you guys will hear about what Avidity is trying to do with that. I believe that natural history studies have developed a few tools to measure the slow progression of the overall FSHD population. And I think that these tools can measure clinical progression in these new clinical trials. And so it's a very exciting time for all of us to see these changes that hopefully will happen soon. And so I want to thank the patients who participated. I want to thank the National Institutes of Health. I think that they are unsung heroes that United States are not treated with the respect and the gratitude that they deserve. And I also want to thank the Friends of FSH Research and FSHD Society for supporting a lot of this research that you guys have.
We may have a question for you. Somebody here wanted to get involved in one of these trials or studies. How would they go about doing that?
Yeah, so I think that we have an email for all our neuromuscular studies to reach out to that. It's also on our website at University of Washington. Our email is, I'll just type it in here. Like that, then you can, yeah.
I'll say your slides. If we have permission to share them later, okay. Any other questions for Dr. May? Oh, sorry. How are you sure? I was interested about the MRI. You want looking at the fat replacement of the muscle over time. Do you ever look at the correlation of that in study with adult onset versus infant onset?
Right, that's a really great question in terms of understanding how the disease is different or similar in pediatrics. I think that that's a hard thing to study because probably a lot of changes before diagnosis, and so how do you follow that over time? I think that the main thing that I would say is that we all know things change, but the thing is you measure it on MRI, and so there's some studies that has been done by the NIH that is both international and this really looking at younger kids and seeing how that changes in the preteen years and teenage years over time, especially using MRIs.
Oh, we have lots of questions. We're gonna go to Eva, you're right. What did you go first?
Thanks, so they'll forget me. Maxed out behavioral follow-ups, if that's the kind that's promising, and you prescribe it off label in the US or Canada.
Repeat that.
So the question is about clenbuterol, whether you can prescribe it off label. I think that's something that we'll have to think more about. In the United States, clenbuterol is not made in the States. It's something that we have to import in from Europe. And so I don't know what the rules and regulations are for Canada. Do you have a sense of that? Especially, I don't know if that's approved or not.
Yeah. If we did due diligence on it, you cannot use it off label because it's not approved by FDA, was the feedback that we got. And it has to do with the manufacturing issue, why FDA that, but it is approved in Europe, so I just came back. So I just wondered how much, there's thought about what we do next.
I think that, so we're running an FDA trial, registered as FDA, and so I think that we can go back to the FDA and tell them about that. And I think what we're really trying to do is trying to set the stage for future study with clenbuterol. So there's definitely some historical precedents for using drugs that have been approved in other countries and bringing it to the United States and trying to get it approved. And a lot of that is driven by certain companies who are just trying to find orphan drugs and trying to develop them. So I think that the point is that we're going to try to improve the concept of whether it works and whether tools that we're measuring can measure the change at the time that we have. And I think that will set the foundation for other people to take it on. And so it's not going to be just 3 sites doing this. It will have to be a lot more, and it have to be probably it would have to be an FDA phase 3 study. It's not a phase 3 study.
Okay, great. One more question and then we'll move on. Great, go ahead.
Yeah, thanks, Leo. I'm just thinking about the math of the reachable workspace score, being an average. And, in that score, you have multiple muscles that are being measured within a population. And so if one person has an improvement in like lateral raise versus horse race, call it, would the metrics that are being used to detect those differences, like could there be signal finding that something anywhere?
Well, I think you want to measure to see there's just improvement. So if you can reach a little bit more, you pull the whole sphere in front of you, then you may be able to do that. I think the issue, it really, at this moment, seems to be when people, people may have aches and pains in certain days, and may be able to do things a little bit differently, and maybe be able to do tests a little differently. So how much noise is it in that? I think that what's cool about it, the most optimal data is that that seems to be a little bit smaller than what I thought it would be. So it seems to have really improved from when we did it was the natural history study to where was still the small one. So hopefully that noise, and you can decrease that noise over time, then you'll be able to see the changes that you have will be bigger than